Hello and welcome to your Chapter 5 video lecture series where we are going to talk about the structure and function of plasma membranes. We'll start off with section 5.1 on the components and structure of the plasma membrane. So our learning objectives will be to understand the cell membrane fluid mosaic model, describe phospholipid, protein, and carbohydrate functions in membranes, and discuss membrane fluidity. So we'll start off with the structure. We know that our plasma membrane is really going to be a border and separate it from its external environment. And in this way, we can control the passage of our organic molecules, ions, water, oxygen, and waste into and out of the cell. We also will have some surface markers that will help differentiate our own self from non-self foreign invaders. For instance, viruses can hijack areas where we have receptors and use it to gain entry into our cells. You can also see these integral proteins that we have, not so much pictured the receptors, but we do have them there, that can transmit signals. So we can either receive extracellular input or intracellular processing activators. So let's talk about this fluid mosaic model. So first it was Hugh Davison and James Danieli in 1935, you can see their model over here, that thought it resembled more of a railroad track appearance resembling a sandwich. So we have the proteins on the outside and the filling would have the lipids. And then in 1972 we had another model that was introduced, and that was S.J. Singer and Garth L. Nicholson, that gave this fluid mosaic model where we see the phospholipids and also some cholesterol, our proteins and carbohydrates that we would find on the external surface. And those carbohydrates would include glycoproteins, meaning we've got a carb attached to a protein, and glycolipids. And you can see examples of these over here. There's the glycolipids, that meaning that the carb is going to attach to a lipid. And they found that the thickness of the plasma membrane is about 5 to 10 nanometers. Now, the plasma membrane makeup is going to vary depending on the type of cell and also the organism. But for a typical human cell, we'll see that the proteins will account for about 50% of the mass, and lipids count for about 40% of that, and that leaves our carbs to be 10% of that. Whereas in other areas, for example, the myelin sheaths that we find around the axon of our nerve cell is going to be 18% protein and 76% lipid. That makes sense because our myelin sheaths are made up mostly of lipid to insulate that neuron. Then we have our phospholipids themselves. So we're really talking about one of these molecules right over here. We've talked about this before too, where we have a phospholipid bilayer. You could see that bilayer over here where the phospholipid heads are on either side and the tails are hidden on the inside. And because we have this conformation or this chemical structure here, we're going to call this amphiphilic, meaning it's dual loving, and the molecule has both hydrophobic and hydrophilic regions. The hydrophobic would be nonpolar, meaning there's no charge, and hydrophilic would be polar, meaning that we have a charge. It is a bit of a negative charge over here. So the other thing I want to mention is that these hydrophilic areas are going to be facing water, of course, since it likes it, and it can form hydrogen bonds and bond with other polar molecules, whereas the hydrophobic area that doesn't like water is going to do whatever it needs in order to hide from the water. So it can form a ball or a cluster in order to keep that area covered up. And these are termed micelles or liposomes. And let's talk further about the proteins that we find within the plasma membrane. These are called integral because they're kind of integrated into the membrane, or they're also termed integrins. And areas where we see it right next to the hydrophobic tails is also going to be an area of the protein that is hydrophobic, whereas on the outside, closer to the phosphate heads, is where we would have the protein's hydrophilic regions. And these proteins are going to come in all sizes. We get a single pass over here that is usually going to have a hydrophobic transmembrane segment that has about 20 to 25 amino acids, but they can of course span a lot larger and can have beta sheets that can make them a little bit wider, whereas the purple here are representing the alpha helices conformation of the proteins. We also have some peripheral proteins. You can see one over here where we find it either on the external surface or the internal surface of the membrane. And many of these are going to serve as enzyme structural attachments for the cytoskeleton or serve as recognition sites. For our carbohydrates, we said we could find these on the exterior surface of our cell membrane. When they are attached to a protein, we call them glycoproteins, and if they attach to a lipid, we call them glycolipids. But collectively, we're going to call them glycocalyx, and they are highly hydrophilic, so that makes sense since we're facing the water side here, and it helps to attract the water since water is attracted to solutes. After all, the term glycocalyx means sugar coating, so water is attracted to sugar. These also serve as markers so that our body, let's say our immune cells rather, are going to come and take a look at it and see, is this a cell that belongs to my body or is it a foreign invader? Hopefully it is one of your cells and it can recognize that and will not trigger an immune response. Now let's talk more about that membrane fluidity. Our integral proteins and lipids are loosely attached to the membrane. So although they look pretty firmly in here, think of it as more wave-like and it being able to create spaces in between so that we can expand and contract. If we had it very rigid, it can burst, and that would of course kill the cell. So what we see is when we were looking at that, let me go back here, the fatty acid tails, we see that one is pretty linear, and this one means that it is super saturated with hydrogen. So we do not have any double bonds here, whereas if you look at the other one, we do have a double bond, which creates a little kink to that fatty acid tail and it kicks out. And that actually helps with the fluidity because as the temperature decreases, we are able to have that kink create some elbow room and some space between these structures. So if we did not have that double bond and both of the tails were linear, then we would see the membrane freeze or solidify, and that would make it more susceptible to rupturing. We also know that cholesterol is going to play a key role in the fluidity because it dampens the temperature effect on both sides. It's going to help to serve as a buffer so that it doesn't matter if temperatures go down or they go up, it will still keep this fluidity to it. And it organizes the clusters of the transmembrane proteins into lipid rafts. And this last slide is just a summary of all of the components that we talked about that create the plasma membrane and their locations.